Good evening, students. So, what about others? So, we'll discuss embryology related to the agronomy. Okay. So, what about others? So, today we'll discuss Purna and Sujata. What about others, ma? Uh,
Hello, sir.
yes. What about others, ma?
Others,
Only,
సార్ గ్రూప్ లో మెసేజ్ పెడతాను సార్ అందరినీ జాయిన్ అవ్వమని.
సరే పెట్టండి, ఒక 2 minutes వెయిట్ చేసి మనం స్టార్ట్ చేద్దాం. ఓకే. So, okay, how many members? So, embryology is the study of megasporogenesis, development of endosperm, embryo, and supports embryological evidence in the solving the taxonomical problems.
Your voice is breaking, sir. Anta clearly it's sir.
Just ఒక్క నిమిషం అమ్మా, నేను ఒక పని చేద్దాం. ఒక నిమిషం ఉండండి, నేను సిస్టంలో కనెక్ట్ అవుతున్నా. So before one day we will discuss in the experimental biology, experimental embryology. So we will covered, I think, my voice is audible.
Yes, sir.
So this is my slide is clear.
Hello. వినపడుతుంది సార్.
Yes. వినపడుతుందా?
Yes sir, వినపడుతుంది సార్.
Yes, anyone respond?
Yes sir, వినపడుతుంది.
Just wait. Okay. So now is my voice is audible?
Yes sir.
So today, according to our syllabus, today we will discuss application of embryology in taxonomy, agriculture and horticulture. So You know embryology. What is the embryology? The evidence have resolved the doubtful systematic position of several taxa. The role of embryology in the solving taxonomical problems was first by the German embryologist in the 1931. So, according to Maheshwari, Bojwani, Bhatnagar, and Radford, some basic embryological characters which have proved to be special basic embryology characters which have proved in the special importance in the taxonomic consideration. It includes presence and type of another tapetum, development of structure, position, orientation of the ovule, megasporogenesis and development of embryo sac, form of embryo sac, type of embryogeny, and fertilization, seed coat. These all are the included in the special importance, the taxonomic considerations. These 8. So, role of embryology. The role of embryology in dicots and monocots. The angiosperms are universally divided into dicotyledons and monocotyledons. The primary classification of angiosperms is based on major embryological characters, the number of cotyledons, right? Clear? So here, helobial type, helobia. This monocotyledons order tetrad as a subclass in some recent systems of classification is characterized by the presence of the helobial type of endosperm. So, next one is archidials. This is the distinguishing embryological characters of members of this order is the presence of undifferentiated embryo and very little or no endosperm. So these are the regarding about archidials. Podostemaceae. The members of this family are recognized because of the formation of pseudo-embryo sac, which is formed by new cellular cells. So, Lemoniaceae. The phylogenetic studies indicate that Lemoniaceae have been derived either from the helobial or from the RAC. But on the basis of the embryological studies, Maheshwari suggested that Lamnaceae have been evolved from the RCAC, not from the helobials. So, Crassulaceae. Embryological study of Crassulaceae suggests it should be placed in the orders of Rosales. Regarding this character, all the above mentioned examples confirmed by the embryology plays a defined and significant role in the solving the taxonomic problems. So these are some diagrams, embryological characters meaning significances of the applications. So the meaning of embryological characters. The embryology, already we discussed what is the embryology, you know, all of you. The embryology is the study of micro and megasporogenesis, gametophyte development, fertilization, development of endosperms, embryo, and seed coats. So it is, although possibility of utilizing embryological characters in the taxonomy was indicated by some earlier botanists like Hofmeister and Strasburger, the role of embryology in solving the taxonomic problems was first brought into the prominence by the German embryologist Schnauf in the 1931. So the embryology characters being less prone to adaptive stress are relatively stable and have acquired great significance in the plant taxonomy, especially when external morphological characters are inconclusive and misleading to results of convergence. So, the embryological evidence have been used in the solving taxonomical problems at almost all levels and have helped the resolve doubtful of systematic positions in the several taxa. However, according to the Johns and Leishinger, the embryological characters have proved to be the significant help determining the relationship in the families, genera, and species. So these studies, it is significant to determining and relationship within the families and within the genera and species, but it is rank of order and also subclass and class. These are so it is very little bit use of the taxonomic studies of embryological approaches. So Maheshwari and John, on the basis of their extensive work on embryology, have provided list of families, tribes, where embryology has either supported earlier classification as proposed new systematic position on the taxon concerned. The embryological characters, meaning, significance, and application. Article, this is shared by the Shashank Goswami. According to these, the article will discuss ecological meaning of the ecological characters, basic embryological character, taxonomic significance, application of embryological data, and some of one article the author shared by the Shashank Goswami is his article will discuss about the embryological characters. What are the embryological characters? The first one is the meaning of the embryological characters, basic of embryological characters of taxonomic significance, and also application of embryological data in taxonomy. So here, the meaning of embryological characters. First one. So embryology, already these are the repeated words. So the plant taxonomists have utilized the embryological characters in the various ways. The analysis of evolutionary and the circumscriptions and delimitation of taxa in making decision of systematic positions. So these are the some characters. The basal, Ariliaceae, Carpelliaceae, Cameralineaceae, Cyperaceae, Mangoliaceae, Nymphaeaceae, Oleaceae, Papaveraceae, Ranunculaceae, Rubiaceae, Umbelliferaceae, Basilateral, these are the Graminaceae. So these are some characters, and also that embryological characters of taxonomy, the importance. Just you see your book page number 288. So according to that, the first of all in this, just all of you see the textbook 288. So anther, the pollen grains, ovules, and the female gametophyte, endosperm, embryo, and seeds, and podospermaceae. These are the approaches to the taxonomical problems. So here the first one is the anther, number of sporangia per anther, 1, 2, 4 or more. Type of wall development, it is basic and dicotyledons, monocotyledons are reduced. So tapetum, the glandular or emboid number and polyploid of nuclei in the tapetal cells. Pollen mother cells, stimulations are successive cytokinesis. Pollen tetrad, tetrahedral, decussate, isobilateral, linear or T-shaped. So next, pollen grains. So number of cells shedding stage 1, 2 or 3. Germpores, number, position and character. So ovules, type of ovules. The arthosporous, anatropous, campylotropous, hemianatropous, circinotropous. So number of integuments, 1, 2 or none. So nucellus, the crassulaceae and pseudo-crassulaceae, teniculaceae. The special features, the presence or absence of endothelium, epistaceae, and sorostaceae, hypostaceae, aryl, cranial, and arbutear. These are the ovules, and also position of functional megaspore and micropolar and chalazal. So next one is the female gametophyte. So embryo sac development, it is mono, bi or tetrasporic. The type of embryo sac, polygonum type, onanthera type, allium type, endymion type, addoxa type, peonia type, plumbago type, peppermia type, brusa type, fritillaria type, plumbagella. So endosperm, type of endosperm, the nuclear and cellular or halobial presence or absence of asteria. The type of embryo, aster, ungrad, unopodiad, and soland, caryophyllid, and pipette. So seeds, the seed coat formed by outer or inner integument. The seed coat surface regulate and reticulate, tuberculate, and fuvulate are otherwise. The some families are marked out of their embryological features. So here, few examples also the caryophyllils. It is more widely known as the centrospermae, and it is embryologically it is very distinct. This order is characterized by the trinucleate pollen, bitegmic and crassulated ovals, which are compilotropous, amphiotropous, commonly with the inner integument and longer than the outer, and very often with the space between the integuments toward the chalagial end. So these are the polygonals and plumbaginals. So these are the licitidales, erichials, genantial cells, and hilobius, archidials, already we discussed in the previous slide. So coming to and podostemacy in the parental aquatic egg. Just you observe the page number 289. So here three diagrams are there. Podostemens, subtulus, and A is the paired pollen grains. In the B is the Trastia, Ramazoinia, longitudinal sectional oval. The C is the mature embryo of Dicrea elongata, showing the prominent suspensor ostoria. So here, some of the addition to it, the following characters are the mark out of this family. Occurrence of pollen grains in the pairs. So anatropous, bitegmic, and nucleate ovals, bisporic embryo sac development, absence of antipodals, absence of triple fusion, absence of endosperm, Solan type of embryogeny, presence of prominent suspensor asteria. So here, Cyperaceae, the family of Cyperaceae, the unique feature in this family is formation of only one pollen grain instead of 4 pollen grains from the pollen mother cell. This single character will mark out the family. The angiosperms, as a rule, each pollen mother cell produces 4 functional microspores, but the Cyperaceae of the 4 nuclei formed after meiosis, only one function during the development of functional nucleus remains in the center, and the 3 non-functional nuclei are cut off on one side of the cell and they do not form pollen grains. The functional nucleus forms in the pollen grain, the other nuclei are degenerated later. So, and next another family is Orangaceae. So Orangaceae, Onyanthera type of embryo sac development is the characteristic feature of the family Orangaceae. So it is not found any other family except as abnormality. The embryo sac is derived from the micropolar megaspore of the tetrad. The 2 nuclei formed after the first nuclear division remain together the micropolar end of the cell. After the second mitotic division, 4 nuclei are formed, of which 3 nuclei constitute an egg apparatus, and the 4th are the functions as the only polar nucleus. So here in 291 page number, just you observe the diagram. So it is in the A is the megaspore tetrad, B is the 2 nucleate embryo sac, C is the mature embryo sac, the functional chalager. So another family is Loranthaceae. This is also page number 291. So the development of endosperm in the Lorenthaceae is unique. In the members of this family, ovals are not differentiated. All the embryos are in an ovary lie close to each other. So and next is the genera marked out by their embryological features. Peonia, the most of the classification genus Peonia is kept in the tribe, the Peoniaceae of Ranunculaceae, but Peonia differs from the other members of the family in vascular anatomy. According to the Worsdale in the 19th century, the floral anatomy, it is different, pollen morphology and chromosome number, and embryology, Peonia deviates from the most genera of the family Ranunculaceae. The most peculiar embryological features of this genus is embryogeny, is embryo development. So here, page number 293, one table is there. Futures and Peonia and Ranunculaceae. Stamen, spirally arranged and centrifugal. Spirally arranged and centripetal. Just to observe that both, what are the difference between these two families? Endothecium, multi-layer in the one layer. Tapetum, mostly two layer, it is only one layer. The pollen, reticulated by the pitted exine, large, elongated the generative cell. Granular, palliplator, smooth exine. You see the page number 293. Small lenticular generative cell. Female archisporium. Multi-celled, many megaspore mother cells function. Unitar, multi-celled, one cell function. Antipodals persist, not polyploid. It is persist except in the Adonis. It is polyploid. Embryogeny, unique and free nuclear, angrad and solan type. Seed, arillate and non-arillate. Fruit, follicle, arihane. So these are the regarding about the difference between these. And also you observe the 294, page number 294, table 19. The futures and Trapa and Ongraceae. Trapa and Ongraceae. So here, same, some difference in the characters. Pollen grains. Pyramidal with six-folded meridional creates, bluntly triangular. The ovary semi-inferior, biolacular with a single pendulous ovals, and each chamber, the inferior mostly tricolor with the many ovals for the chamber and axial placental. Embryos are polygonal type, onanthera type, endosperm absent is the present. Embryo, solan type, ongrad type. Suspension, well-developed suspension in the Hastoria, in the Trapa, the short end inconspicuous. Cotyledons, one cotyledon extremely reduced. In Orangaceae, cotyledons is equal. Seed coat. Testa multiplicative, testa not multiplicative. Fruit, one seeded drupe and lucidal capsule. So these are the regarding about the some taxonomic evidences in the embryology. So these are the already covered. So next we will applications of embryology in the agriculture practices. So just wait.